Hello students, today we are going to learn about the genetic code. What is the code which is coding for a particular protein? How do protein comes to know that what is the information which is there in the RNA? Because we have seen that RNA has taken the information from the DNA in the process of transcription. Now the genetic information lies with the RNA. Now from RNA, this information has to go to the protein so that the protein can be formed. The amino acids will join together to form the required protein. But how that is done? There must be some code which is going from RNA to amino acid so that the specific kind of proteins can be formed. Now, what is this RNA? What is this code, and how many nitrogenous bases does it contain? This all we are going to learn in this video of ours. Genetic code. Now, genetic code is the code which directs the sequence of amino acids. Now, the information is with the RNA. So, RNA will give this as per the central dogma. The RNA is going to form the proteins. But till now, there was complementary base pairing. From DNA, the RNA was formed because of the complementary base pairing. But now, there is no complementary base pairing in proteins. So, how proteins are formed and how do the different types of amino acids come to know that which amino acid has to join with which amino acid to form what type of protein? So, here there are codes, and this is called the genetic code, which directs the sequence of amino acids during the synthesis of protein. Now, George Gamow, he was the scientist who proposed that if 20 amino acids are there, total 20 amino acids are there, and with those 20 amino acids, all proteins are formed. For example, there are 26 alphabets in your English, but the whole English is whole letters, whole sentences are formed from those 26 letters. Similarly, in chemistry, 20 amino acids are present, and from these 20 amino acids, all proteins are formed by different combinations. Clear? And we are having this George Gamow said that we are having 20 amino acids, and these 20 amino acids are to be coded by 4 bases because RNA is having 4 bases. What are these 4 bases? Adenine, guanine, cytosine, and uracil. So 4 bases are present, and these 4 bases are going to code for 20 amino acids. So there has to be combination of bases, because if there is one base for one code, so one base for one amino acid, so only 4 amino acids can be formed. And if there are 2, only 8 amino acids can be formed. But we want at least 20 amino acids are there. So he proposed that the code should be made up of 3 nucleotides, and it will give 64. 64 means more than, it is much more than 20, but it was a bold proposal by George Gamow. He said it can't be 2 because with 2 bases there can be 16. By permutation and combination there can be 16 combinations, but if we are taking 3 together there are 64 combinations, and 64 are the, so he said the genetic code is made up of 3 nucleotides. Now the amino acids are 20 and genetic codes are forming with the 3 bases 64. So it was very difficult for many scientists to digest, but then afterwards with several other experiments it was found to be correct. So the codon was proposed to be a triplet, 3 nucleotides in one genetic code. Hargobind Khurana, our Indian scientist, He developed a chemical method to synthesize RNA molecules with defined combination of bases. What he did, he developed a scientific chemical method in which he synthesized the RNA molecules with different combinations of bases. And similarly, the Nirenberg was the another scientist who developed cell-free systems for protein synthesis. Without the cell, under artificial conditions, he synthesized the protein with the help of the codes. And he also took the triplet code and how it can be deciphered, how it can be read by the amino acids. Now, the enzyme known as enzyme. This is a polynucleotide phosphorylase. This helped to polymerize RNA with defined sequences in a template independent manner. That means there was no template. Without the template, the RNA was synthesized, RNA was polymerized, RNA was made with the help of this enzyme. So, this all happened in the laboratory, and with all these experiments, the proposal of the George Gamow was found to be correct, and a checkerboard was formed. And this checkerboard finally gave the genetic codes. Now, what are this checkerboard? They are the different combinations. We are having four nitrogenous bases in RNA. So it is uracil, cytosine, adenine, guanine. Uracil, cytosine, adenine, guanine. Now at third position, you write all four together. So these are the different combinations which can be formed. So total 64 codes can be formed if we are taking 3 together. So with 3 together, these are the 64 codes which are formed. Now, what are the features of a genetic code? Codon is triplet. So triplet means it is having 64 codes can be formed from 4 bases. And out of these 64, 61 codons are coding for amino acids, whereas 3 are not coding for any amino acid, and these 3 are stop codons. They are called stop codons. Now, one codon codes for a specific amino acid. Now, these are the properties of a genetic code. One codon is coding for one amino acid. That means if GUG is there, GUG will always code for valine. It will not code for any other amino acid. So this property of codon is called unambiguous. So first property, first feature of genetic code is it is unambiguous. Unambiguous means there is no doubt about it. So it is one is coding for one specific amino acid. So this is unambiguous. Second property of codon is that these codons are degenerate. Degenerate means some amino acids are coded by more than one codon. That means, see here, this valine is coded by GUU, GUC, GUA, GUG. That means all 4 are coding for valine only. So this property is called degenerate. Second property of codon is that they are degenerate. There are some amino acids which are coded by more than one codon. One codon can code only for one specific amino acid. Many same amino acids can be coded by one codon. More than one codon are coding for same amino acid. So this property is called degenerate. Unambiguous, degenerate. Third property is genetic code is universal. Universal means if that code is present in bacteria or in tiger or in human being, it will code for same amino acid in all species. In all species, it will code for same amino acid. For example, this GUU is coding for valine amino acid. So it will code for valine amino acid in bacteria, in dog, in tiger, in frog, in human being, everywhere in all species. This code will code for the same amino acid, and that property of genetic code is called universal. So three properties are there of codon. One is unambiguous, second is degenerate, and third is universal. So these three are the properties of genetic code because they are very, very specific. Now another thing is that codons are read continuous. They lack punctuation. Like if we have to read a genetic code, we will not say A and UG. No, we will say AUG, all three together without any punctuation. So they are read continuously. And even on segment, a strand of DNA or RNA, they are found and they are present in continuous manner. There is no space after any every codon. There is, you will not find any space after AUG and then some other codon. No, they are continuous, but we have to read them continuously. Three are read, and these three are read together, and they otherwise they lack punctuations. Now AUG is a very special codon which is having two functions. This is called a start codon. As I told you, out of 64, 61 are coding for amino acids, while three are not coding for amino acid, and they are called stop codons. Out of these 61, one, which is AUG, it has a dual function. It will start, it is acting as a start codon. So everywhere, wherever the translation process is taking place, the start codon is always AUG, and at the same time it is coding for methionine amino acid. So it has got dual function. One is it is coding for methionine, and second is it is acting as a start codon. So this is about the genetic code. Now we will see, these are the amino acids. We have 20 amino acids, and these are the symbols of amino acids, and these are the codons which are coding for this amino acid. For example, alanine. Now, alanine, the symbol is ALA, and what are the codons? GCA, GCC, GCG, GCU. All four are coding for alanine. So this property is degenerate. Many codons are coding for alanine. Unambiguous because GCA will never code for cysteine. It will always code for alanine. This property is unambiguous. Third property, universal, is alanine. Even in bacteria, this will code for alanine. Even in any other species, this will code for alanine. So these are the 20 amino acids, and these are the symbols, and these are the codons which are coding for them. And this AUG is the special codon which is called start codon, and it is starting the process, and at the same time it is coding for the amino acid methionine. So we have seen that genetic code is a triplet of sequence which is coding for one amino acid. Now then these amino acids will join together to form the required protein. Now we will see what are the effects of mutations on genetic code. We have done mutation in detail earlier. You can refer that video of mutation if you want. But here we will see how the genetic code is changing because of the mutation. Now, mutation can be insertion, can be deletion, can be inversion, can be rearrangements. So these all things happens in the mutation. Now, mutation is what is the result of the mutation? It is changing the phenotype of the, because of the change in the genotype, it is changing the phenotype of the person. Now this phenotype, for example, sickle cell anemia, we have done sickle cell anemia. In sickle cell anemia, glutamic acid was changed for valine acid because of the change in the coding for beta globulin chain in hemoglobin. So there was only one change in the genetic code, and because of that one change in the genetic code, what happened? In spite of forming glutamic acid, the amino acid which was formed was valine. Change in the triplet in the genetic code, and that changes the amino acid which is formed. So because of the change in the base one, change in the genetic code, in spite of formation of glutamic acid, the amino acid which was formed was valine. So that changed the structure of the beta globulin chain, and this results in the sickle cell anemia. These mutations, which are due to the single change, they are called point mutation. And insertion or deletion of a single base pair disturbs the entire reading frame in the messenger RNA. This also we have done in detail. You can refer that video, and that is called, it will change the complete frame. Before the deletion, it will remain the same, and it will change the further many frames. So that's why it is called frame shift mutation. Because one single base pair is disturbing the entire reading frame. Why? Because we have to read 3 3. So if we are reading 3 3, the change will be there in further all three. If there is change in one deletion is there or one addition is there, it will change the whole frame, and that is called frame shift mutation. Now, frame shift mutation hold the proof of the fact that codon is triplet. Now, this again, frame shift mutation again is a proof that codon is in triplet, because if we insert 3 or multiple of 3 bases, then followed by the deletion of the same number of bases, the reading frame will remain unaltered. If we have added 3 3 together, and if we have deleted 3 3 together, then the reading frame will remain unaltered, because there will be no change in the reading frame. So this again proves that the codon is triplet. The properties of genetic code.